Good afternoon, friends. Hello and welcome to the EduSat Network. Our topic of discussion today shall be a continuation from the previous one, where the subject expert discussed the biography of Mendel and his discovery. And friends, today's lecture's topic will be Transmission Genetics, Mendels Laws Revisited. And with this, I would like to welcome our subject expert to our studio, Dr. Eklavya Chauhan, Associate Professor, Department of Botany, Deshbandhu College, University of Delhi. Dr. Chauhan has been teaching for nearly 4 decades, and with this brief introduction, I'd like to welcome him and also request him to begin our today's lecture. Thank you for coming, sir.
Thank you, Urvashi. What we discussed on the last turn was a brief and not so brief biographical sketch of the father of genetics, Gregor Johann Mendel, and his works. And partly we also discussed how this work went into oblivion, and finally, 16 years after his death, the whole work was accidentally, as a matter of pure serendipity, it was rediscovered by the 3 scientists who were magnanimous enough to recast the entire work and put it together under the banner Mendels Laws. Today we will discuss in details the Mendels Laws. Why I chose the topic as Mendelism Revisited is that after the classical discussion of Mendelism, we would also like to just view the entire Mendelian expression in terms of the modern perspective. What does the DNA profiling or the electrophoresis say about the wrinkled seeds or the other characters? So let's begin with a little recapitulation of what we discussed on the last turn. If you remember, Mendel had chosen 7 characters, and they were purely as a matter of chance that he chose these characters. The only thing perhaps that these characters had in common was that all these 7 characters were very, very sharply contrasted. I had also pointed out, and we had discussed in details, that out of these 7 characters, it was later found out, many years later, that these particular 7 characters, the genes, were located only on 4 chromosomes. That is on chromosome number 1, chromosome number 4, 5 and 7. And in particular, the chromosome number 4 deserves a special mention, because if you can observe, it carries 3 genes, 2 on one side of the centromere and 1 on the other. The character of pod shape, which means the fruit shape, and the plant height, they are so closely packed together that Mendel could have never got the desired results as he was contemplating. So literature says that he never mentioned the results of these crosses. We have also discussed that these 2 genes would be definitely linked genes and would not fit into the notions of Mendelian results. We could discuss these particular linkage results a little later in one of the next episodes where we have linkage and crossing over. But it would be in the fitness of things to start with the terminology of genetics. As I said before, a good mechanic would definitely like to have a knowledge of all the parts of a particular car. Then only it would be possible for him to assemble and disassemble the entire machinery. So as the story begins, Mendel never knew the term gene. His biography says, and some of his works which have been preserved now say, that he had always, at time and again, used the term Anlage, which means that there was some sort of a predisposed factor, or he also at certain places has used the term factor. Correns, who was one of the rediscoverers and was given the responsibility to frame the laws, has also used the term elemente. Now these 3 terms, Anlage, factor and elemente, point towards one and the same term, which is gene. So we could also define genetics as the study of genes. The term gene, as you can see, came much later in 1909, a little after the laws were formulated, and Mendel was already established as the father of genetics. So a gene in its classical sense could be defined as the smallest structural and functional unit of heredity. Friends, as I told you before, one has to be very particular about the calendar in such type of discussions. This is a story of 1909, and today our concept of a gene would be, if not a little different, but definitely much more pervasive in our view. It was Beadle and Tatum, the famous duo, who are also Nobel laureates, who worked on the Neurospora crassa, the fungus, and they worked on the auxotrophs, that is the nutritional mutants, and they came to a conclusion that one gene was in fact responsible for the synthesis of an enzyme, and this is how the one gene, one enzyme hypothesis came. We are actually looking back and tracing the entire history as to what is our present conception of a gene, because we started that this is the smallest, and therefore we committed ourselves to say that it cannot be further divisible at all. But then that was 1909, and we are past a century since then. Janowski and Ingram were two biochemists who investigated the whole process a little in details, and they came to a conclusion, okay, enzymes are made up of proteins, and the proteins of polypeptides, but then sometimes it so happens that a particular enzyme is so complex that it is made up of more than one polypeptide, and these two polypeptides are controlled by two different genes. So they said, why not modify the whole idea, saying that one gene, one polypeptide hypothesis. If we look at the least common factor in the above two hypotheses, we would find that nobody has touched the gene so far. That still remains indivisible, whereas the enzymes have been further classified into one or more polypeptides. So it had to wait till the 1960s, and if I remind you, 1960s was one of the most fertile decades of molecular biology, where very important discoveries were made in terms of transcription and translation. And the Frenchman Simeon Benzer in the 1960s was the first person to have suggested that the gene which was hitherto indivisible was actually in a position to be classified, to be broken down, or to be divisible into three different entities. He introduced the term cistron, which was the functional unit of a gene, a recon, which was the smallest unit which could undergo recombination, and finally the muton, which was the smallest unit, if when changed would cause mutation. So immediately the whole scenario changed. Gene which was hitherto indivisible could be further divided, and it was therefore no longer the smallest unit. And by that account, muton seems to be the smallest of the units. So that means now we know that we have monocistronic and polycistronic genes, and one gene in many cases could be composed of many cistrons. So that means we are not undermining the importance of a gene, but we are only trying to look at the magnitude that the gene possesses. So finally, the earlier two hypotheses, they were further modified into the present day one cistron, one polypeptide theory, which holds good for quite some time now. Let us think of ourselves. We are human beings, and our chromosome number is 46. We always talk of a diploid, which means 23 of our chromosomes have come from our father and 23 from the mother. So each chromosome coming from one parent should be having a pair or a counterpart which is coming from the other parent, and therefore these are called as the homologous pair of chromosomes. One should remember the term homologous is distinctly different from the term homologous, which we would discuss at the right moment. Now that means each chromosome would be having various sites at which the genes are located, and these particular sites would be called as the locus, the plural being the loci or the loci. So that means, for example, if at a particular point there's a gene for say hair texture, whether it is curly or straight, that means the other chromosome which is having the same gene or a different one should be at the same locus. So that means this brings us to another question. Can our hair, for example, be curly and straight at the same time? No. But then there's a possibility that it could be either of the two. So the term allele seems to be a very important one. The term was given by William Bateson, who gave the term genetics also. In 1902, he also talks about allelomorphs, or the organisms having these two alleles or allelomorphic pairs. So by definition, an allele is nothing but one of the counterparts or one of the two alternative forms of a gene which are present on the same locus of a pair of homologous chromosomes. In other words, if we are talking about the character of height, it could have two alternatives, tall and dwarf. We are not talking about ourselves because we don't come into the category of tall and dwarf all the time. We are somewhat intermediate. So our height is a case of polygenic inheritance, which would also come subsequently. But then right now we are restricting ourselves to a discussion of sharply contrasted characters, which means tall and short. So that means the character is height, not tall or dwarf. These two which I mentioned are the alternatives of the character. So this is how we distinguish between a character and a trait. In other words, a character would be a well-defined morphological or a physiological feature. For example, what is the color of a flower? What is the height of a plant? What is the color of a seed? What is the texture of a seed? So these are characters. But then what kind of color does it have would be its trait. That means a trait would be a distinguishing feature of the character. For example, dark, it is light. So these two are the two alternatives of the same character. And tall or dwarf. So that means each allele by definition is going to control different expressions or traits of the same character. It is very, very true and a matter of common sense that a gene is not going to express both its alternatives at the same time. It would either be tall or it would be short. It would either be dark or it would be light. A pictorial depiction of Mendel's traits means that, for example, restricting ourselves to garden P, if we have an allele R, we'll talk about the capital and the small in a minute. So that means this is defining the character of seed texture. Now the texture could have two alternatives. Capital R would mean that is the locus or the two levels. The two levels, as you can see, are the same on a pair of homologous chromosomes. One of them is a paternal and the other one is a maternal chromosome. So that means these two different alleles of a gene, they are going to occupy the same locus. And the small r would signify that the character signifies a trait which is of wrinkled seeds. So that means these alleles are controlling different traits of the same character, which again brings us to one character and two traits. Subsequently, our subsequent discussion will tell us that there are multiple alleles also. For example, our blood groups, they are characterized by multiple alleles. Then comes the age-old terminology of phenotype and genotype. We always say, you look like what you are. Why don't we say, what you are is reflected in how do you look? So that means the term, these two terms were given by Johansen in the year 1911. So any external manifestation or any external appearance of a character of a particular plant or an animal is called as its phenotype. For example, somebody's skin is tanned, so we say, oh, the fellow is a bit dark. The other one is light, so we say it's one has a fair complexion. The flower is having a red color or a purple or a white one. So that means this is how an organism looks like. So whatever are the characters, tall, dark, wrinkled, smooth, they are all external characters and they are called as phenotypes. One should not forget, the phenotypic character of an organism may also change temporarily. Look at ourselves. If we are going to spend or toil too much in direct sunlight, we might get a lot of melanin and may turn dark. And then finally, if we are remaining in the air conditioner for a long time, we could perhaps get a little fairer. But then it's only a temporary change of the phenotype. So as to suffice here would be that environment plays a very important role in modifying the phenotype. But then it would be incomplete discussion without what is there in the genes, which means the genetic constitution of an individual as expressed in terms of certain symbols would be its genotype. So that means a phenotype is nothing but a manifestation or an expression of its genotype. Let's rephrase our earlier statement. We look what we are. Of course, the environment modifies this whole concept. Once we have said that we have the dominant and the recessive of the traits, and I mentioned that out of the two traits, only one would be visible at the same time. Now the question is, which one? On a pair of homologous chromosomes, if both the alleles are of the same type, then they would be called as homozygous. The term was given by Bateson and Saunders. If the two alleles are different, I gave you the example of curly hair and straight hair. Now which one prevails comes a little later, but then the genotype shows that we have both the alternatives in a homologous chromosome pair. It will make a heterozygous constitution, genetic constitution. This term again was coined by Bateson and Saunders, but then heterozygous in common terms would mean impure, and an impure is nothing but a hybrid. If by chance there is a mutation or any other such event, it takes away one of the alleles. So that means only one of the alternatives is there and the other alternative is not there at all. But that particular locus is empty. And naturally, when there are just one alternative, then whatever that alternative is there, it would be expressed. So we will talk about the front wheel and the rear wheel only when we have two wheels. If we have just one in our hand, so what is rear and what is front? We can't make it out. And likewise, if something happens that in a sense that both the alleles of a character are missing, that means that character would not be manifested at all. In that case, it would be an azygous condition. Now the question comes, what if both the alleles of the same character are different. Which one should prevail? So the one which prevails is naturally the dominant one. And a dominant allele by definition is one which will express itself in any situation. By any situation we mean in a homozygous condition, or in a heterozygous condition, or even in a hemizygous condition. Obviously, there will be no expression of any trait in an azygous condition. So that means regardless of the presence of the other alternative allele, this dominant allele would express itself. So a recessive allele by definition would be therefore that character whose expression is manifested only when it is in a homozygous condition. Well, there could be an alternative where there is a recessive allele and there is no dominant allele to dominate over it. So that means this would be a condition which would be called as a hemizygous condition. The recessive allele would definitely express itself in a hemizygous condition. Those of us who are familiar with the X and Y chromosomes, we know that any character, any allele which is present on the X chromosome of a male, whether it is dominant or recessive, actually many of the diseases which are X-linked in human beings are recessive. All of them would be expressed in a male in a hemizygous condition, and in a female they could either be dominated by the dominant allele, because there are two X chromosomes to a female. We will talk about this when we are talking about sex-linked inheritance. So that means before starting any discussion, we need to give certain symbols to the dominant and the recessive. So it's a convention to designate the dominant alleles by capital letters. So that means if we are designating tall, we would have a capital T, and likewise small t would represent a recessive allele. If we have capital T, that means we are talking about homozygous tall or pure tall. Small t would mean it's a homozygous recessive, and naturally it will be small. But then what would happen? Capital T and small t together would be a heterozygous genetic constitution genotype, and it would go according to what the capital T says, because it is a dominant allele. And of course, small t and nothing means the other alternative site is vacant, and that would now go according to the character of the recessive one. In the absence of a dominant allele, when a recessive allele is expressing itself, it's not true dominance, we know. So another term has been given to it, and that is called as pseudo-dominance. It is seen in nature that the wild alleles which have been which have come as a result of the testing of time and the natural hybridization, they are usually dominant. And the ones which have arisen as a result of mutation, they are usually recessive. So there is another terminology for mutant genes. We can use a term plus also to designate these particular characters. So just to summarize, Mendel, who died in obscurity, could not formulate the laws, but then he was in a position to give his postulates on the basis of the statistical data that he had collected. And the first important postulate that he gave was the principle of paired unit factors. The modern concept we have just studied in details is nothing but the alleles, which are the two factors of the same trait of the gene. He also gave the second postulate that when both of these factors are present, only one would prevail, the other would be masked. The one which prevails is dominant, and the one which gets suppressed or remains suppressed is recessive. However, there is no blending at any time, at any point of time, because anything which is suppressed does not mean that it gets mixed up. It is only suppressed for the time being and may show itself or express itself in the next generation. So that means he was very clear about the principle of purity of gametes. That means each of these factors, they would keep their identity up to the last generation. And finally, no matter how many characters we take, these particular characters would be inherited in the next generations independent of each other. If we look carefully, the postulate 3 and 4 are the ones which really got transformed into the two famous laws of Mendel, namely the law of segregation, the first law, and the second law, which was the law of independent assortment or the law of random hybridization. Of course, they were formulated by Karl Correns. Looking at the graphic representation of the crosses of Mendel, because Mendel's crosses were based only on the phenotypes, and what his results were formulated were all phenotypically based. But then one has to now look at the level of the genes and of course the modern DNA concepts, which we will come. So in the parental generation, if we take say two phenotypes which are tall and dwarf, as one can see they are pure. That means the homozygous dominant and the homozygous recessive. Then we get the F1 progeny as capital D and small d. They are heterozygous, but then all the progeny of the F1 or the first filial generation are all tall. So that means the principle of dominance stays. When members of the F1 generation are allowed to self, that means now the genotypes of the parents capital D, small d are crossing with one another. They would produce two types of gametes, and these two types of gametes can now cross with each other, and this crossing can be represented by what is called as the Punnett square. We are familiar with the term Bateson and Punnett, whose name would appear many times when we are talking about the linkage hypothesis and the coupling and repulsion hypothesis. So this is a checkerboard which they made. That means this is one parent and this is the other parent with their gametes, and we have now the phenotypes of tall, then two hybrid tall and one dwarf. So that means the F2 results show that the phenotype is 75% tall and 25% dwarf. So that means this is the phenotypic ratio of 3:1. Mendel's work was only till here. That means on the basis of this result, Correns formulated the first law or the law of segregation. That means the genes, they segregate in a phenotypic ratio of 3:1 in the F2. But in today's perspective, we can see that the genotype is pure tall 1, 2, that is 2 by 4, that is 50%, hybrid tall, although we can't make out by the faces of the plants that they are hybrid or tall. We'll talk about it, how to know the genotypes and what type of crosses to perform, and 1 dwarf. So that means the genotypic ratio happens to be 1:2:1, which is the genotypic ratio for a monohybrid cross. The methodology we have discussed earlier. No matter whether we take a true breeding round and make it into a female by emasculation, that is by the removal of all the anthers, and we cross it with a true breeding wrinkled seed, that is the flower coming from this seed, and then use it as a male by dusting the pollen grains. No matter how many times we perform this experiment, all the plants in the next generation are going to have seeds which are round. So that means this is simply because of the fact that both the parents that we have taken are homozygous. That means they are pure. And what Mendel also tried was the reciprocal cross. He said, no, let me just reverse the parents. And he took now the flower of a plant whose pollen grains were of a wrinkled variety, and when he crossed, he found that all the seeds in the pod were still round. And if the ovary was taken from the wrinkled variety, still the same results. This is a reciprocal cross, which means no matter whether you take a female coming from a round seed or a male coming from a round seed and cross it with the wrinkled seeded other sex, you will always get the round seeds at the end, simply because of the fact that the round seed character dominates over the wrinkled one, provided both the parents are homozygous or pure for the character. Let's look at this discussion with the help of the allele positions. If we designate the seed shape as capital S, that means capital S would denote the round ones and the small s at this locus as the wrinkled one. Then we have the dominant allele. It still is dominant in a heterozygous condition. In a recessive condition only, in a homozygous condition, would we find that the seeds are wrinkled. Otherwise, they would ever remain round ones, which again signifies the fact that the capital S allele is going to express itself regardless of the presence of the other allele. So it is completely dominant. We have many instances which we would study subsequently, that this dominance of the alleles may not be complete in all the cases, and we have complete instead of complete dominance, we have incomplete dominance. A classical example would be in the sweet peas or Lathyrus odoratus, where when we cross a red flower with a white flower, we get ultimately the plants which are neither red or white, they are pink. So we will discuss whether it is still a case of blending or something else. It's a case of incomplete dominance or partial dominance. But by and large, all the characters which Mendel selected were all completely dominant. Looking at the concept of dominance and the principle of segregation, these are the results which bring us to the 3:1 ratio of the F2 phenotypes. Mendel was meticulous enough even to go to the third generation, that is the third filial generation. One can find that those gametes which are pure R and R, they would always give rise to a round seed. The ones which are having both type of gametes, because they are coming from the heterozygous capital R and small r, if they were self-fertilized, they would still give round and wrinkled in a ratio of 1:2:1. And the ones which were wrinkled, which were having both R and R, there was no question of having any dominant character whatsoever. So that means the homozygous wrinkled peas, they produced only wrinkled peas. So that means, how do we know that when we are talking about this 1:2:1 ratio? I mentioned earlier that by their faces we cannot say whether a particular organism is a hybrid for a particular character or a pure one. We'll have to devise some other means. So that means, how do we know, or how do we distinguish a hybrid from a pure character in an organism? For this we'll have to make some crosses. A cross between the F1 progeny and any of its parents, whether the dominant or the recessive one, would be called as a back cross. But if a cross with the F1 progeny made strictly with the homozygous recessive parent is called as a test cross. It is this particular cross which is providing an evidence. It is providing a testimony to the genotypic status of the F1 progeny. We cannot a priori call this F1 progeny as a hybrid, because that is our problem. We have to find out what this progeny is. Is it pure or is it a hybrid? If it is a hybrid, then in the back cross, which is now a test cross, it would show always a ratio of 1:1. One thing is very clear, that all test crosses are back crosses, but not all back crosses are test crosses, because test cross has to be only with the recessive parent, and we will get a 1:1 ratio, indicating that it is a hybrid which we are looking at in the F1 generation. If we try to compare a homozygous or a pure set of parents to the parent which is a heterozygous one, then we would find how the situation goes. A pure parent, both the pure parents in the monohybrid cross would have all the characters in the F1 generation as the dominant ones, and we would now be sure that the parent were pure, because there is no alternative. It's all pure all the way. If we were taking the smooth seeds and a heterozygous parent, we would find that the results would not be the same. We would find at the F2 generation that half of the seeds of the plants are smooth, whereas the other half are wrinkled. So that means the smooth and the wrinkled seeds, they have been segregated in the ratio of 1:1. That is an equal proportion. Now this makes us sure that the parent that we took into consideration was a hybrid and not a pure one. So in order to conclude, Mendel's first law, it would suggest, and that is the law of segregation, which is nothing but a manifestation or a modification of the law of purity of gametes, that the two members of a gene pair, that is an alleles, they segregate or separate from each other while they are forming the gametes, and so one of the allele would go in half of the gametes and the other allele would go in the remaining half. So you could also appreciate that it's a matter of chance as to which one fuses with which one in the next generation. If we could look at a cytological basis of this whole cross, one should be very clear that the process of crossing over is a must, because we are always talking about which one is going to cross with which one, which means there has to be a reshuffling of these alleles at the time of gamete formation, and then only they would be crossing with each other randomly. Another question which bogs our mind always is that, is the ratio 3:1 always associated with the Mendelian phenotypic F2 ratio, which is the basis of the law of segregation, absolute? Which means, does 3 mean 3.0 and 1 mean 1.0? Certainly not. If one was to look at the statistical account of all these crosses, one would appreciate that the ratio ranges from, say, 2.96:1. Sometimes it is even more than 3, which means 3.01, and the highest in terms of the flower color, which is 3.15 versus 1, and as low as 2.84 versus 1 when we are talking about the plant height, on which most of our crosses are based. So that means when we are talking about 3:1, one is looking at an arithmetical mean. This is where Mendel scored, because in the first episode we came to a conclusion that it was the statistical mind of Mendel and the analytical power which really prevailed, and which was of a great advantage, where he could score over his predecessors like Wolff and therefore all the characters would never give an absolute value of 3:1. It is only an average, and he expected this. So that means he also realized that these results were a part or within the range of statistical error, and therefore there could be broadly the whole trend could be appreciated as, say, 75% versus 25%, that is 3:1. Let us now look at the modern perspective of what this whole business of the genes in terms of wrinkled and the smooth character is concerned. Why? Because we find that if the seeds are round or the seeds are wrinkled, that means there must be some difference in their DNA. We've already discussed in terms of their molecular weights, or we have even designated them by, say, capital T or small t, but in this case as capital S for smooth and small s as wrinkled. In the modern context, the DNA analysis is of paramount importance. Actually, the smoothness and the wrinkled nature of a seed is due to the presence of an enzyme, which is the starch branching enzyme, which is designated as capital SBE1. This is a gene which is present, and if it is present, then it would always mean that this particular gene would have coding for the starch branching enzyme, which would now make a branched chain compound, which is amylopectin, and amylopectin would be responsible for uniform shrinkage, and the seed would appear as smooth. If there is a transposable element, a transposon, which can somehow break the DNA into the normal W form, the capital W, which is the smooth one, and it can insert itself, number one, it will make this whole DNA heavy. When it makes the whole DNA heavy, then the electrophoretic properties would also change, and this would suppress the SBE gene 1, and as a result the starch branching enzyme, that is the amylopectin, would not be formed, and there will be a non-uniform shrinkage which would result in a crumpled or the shriveled or the popularly called wrinkled seeds. So this is the basis. How do we actually demonstrate and prove that this whole hypothesis is correct? Because the electrophoretic properties, if we are going to take or extract the DNA from these genes and put it in an agarose gel, we can see its electrophoretic property on the basis of the pattern of bands that it shows. I have just told you that if there is a transposon entering the sites closer to the capital W, it would make the whole DNA as a larger one, and the larger the size of the DNA fragment due to transposable element, slow would be its movement on the electrophoresis. The capital W by and large is smaller, and therefore it is a fast moving one. One can see the electrophoretic bands on an agarose gel and just try to interpret the classical Mendelian characters in a new parlance or in a new terminology. It would mean that the capital W homozygous situation, which is there for round seeds, is going to have just one fast moving band, because this is a band which is without any transposon. And the moment you find that there is a wrinkled one, that means we'll have WW only, and it is a band which is slower moving. The heterozygous would show two bands. So we will immediately come to know that it has one capital W and a small capital W. So this is how we could represent a electrophoretic pattern. If we have a parent which is capital W, it is faster moving, it goes down. We have a small w, then it is slower. So we have the same gametes, we are interpreting the same monohybrid cross, and in the F1 generation we have now two bands in the electrophoresis agarose gel, and it means that the WW parent is now going to, in the next generation, produce an equal number of gametes. So that means these particular nature of the electrophoretic pattern also translates ourselves into the modern terminology, which means if we are talking about 1:2:1, we say that there is one band here, there are two bands here in both of them, and the one band for small w and small w, which is ultimately giving rise to the zygotes of 1:2:1. That is 1/4, 1/2 and 1/4. So this is the modern interpretation. If we were to take the statistical account of the electrophoretic pattern or the DNA images, we would find that depending upon which of the parents we are taking from the seeds, we would have the 1:2:1 situation. So we can prove it either way, either the classical way or in the modern context of the DNA electrophoretic pattern. Likewise, if we were to signify the test cross, see, we have a heterozygous parent, but we don't know. But then we find that there are two bands. So one signifies capital W and the one above signifies small w. So we are sure that we are talking about a heterozygous one, and this is not a pure strain. And then this would give rise to two types of gametes, and ultimately we will have one half and half, which means 1:1 ratio of the test cross. So that means the 1:1 ratio can also be easily proved by these means. So that means the wrinkled seeds paradox has been demonstrated more than 100 years after Mendel's classical experiments of the garden pea. So it took almost a century to come to a conclusion as to what was the underlying enzyme which was responsible, and this gene is designated as capital S, capital B, capital E1. Let's then come to the second law. Now Mendel was very happy and satisfied that his results were giving a tangible direction of 3:1. The next idea which struck him was that I have taken just one character at a time. What would happen if I were to take two characters at a time? Will these characters get mixed up? If not, which ones would be seen and which ones would not be? And what would happen to the pattern of their expressions, especially the ones which are not to be seen in the next generation? Would the expression of one of them be influenced or would influence the expression of another, or would they be inherited independently, or would it be a random process, or there would be a particular distinct pattern? So that means he now took two characters, and the classical experiments which he performed was again on the color as well as texture of seeds. Hitherto we have seen only the color of seeds, the texture, that is the smooth and the wrinkled, the round and the wrinkled. But then now he took the additional character of the color. We find that the yellow color of seeds are dominant, the yellow character over green. Now one would immediately ask, we have not seen many of the peas which are sold in the market which are yellow, we always eat green. Now, this is a fundamental question of dominant and prevalent. Any gene which is dominant need not always be prevalent. I always give my students an example as to the curly hair is the trait is dominant over straight, but most of the people which we see around us are straight haired, isn't it? And it's only the beauty parlors which can make, or a hair treatment which could make a person curly from straight hair and vice versa. But by nature we find that the curly hair character is dominant, but then we have less. So that means there's a difference between dominance and prevalence. This also we will discuss when we are discussing terms like penetrance, expressivity, and of course the prevalence. So that means these two characters were now taken, and now one would have two genes, capital R, capital Y for round and yellow, and another homozygous parent which is a double recessive, we call it in parlance, would be of the wrinkled as well as green seeds. Please don't forget, Mendel was sure that the dihybrid cross that he was making was nothing but a set of two monohybrid crosses, because these two characters were there, and therefore there had to be some relationship between the results of the dihybrid cross and the monohybrid cross in terms of calculations. So when he saw the first generation, all the seeds of the F1 generation were round as well as yellow. So that means the principle of dominance prevailed. When now the gametes were formed, there would be four forms of gametes. Immediately we would say we had two types of gametes in case of monohybrid cross, and we have four. So that means there has to be some relationship like 2 to the power 2 or 2 into 2. We will look at it at the end. So when four types of gametes are going to cross with another four type of gametes, that means naturally we would have 16 combinations. So now there is again some relationship between 16 of the dihybrid F2 and the 4 of the monohybrid, and it happens to be a square. And also let us look at the phenotypic ratio. The phenotypic ratio here is 9 round yellow, 3 round green, 3 wrinkled yellow, and 1 wrinkled green. What does this whole idea mean? It means that the character of roundness is inherited independently of the character of the color of the seed. In other words, the yellowness of a seed could very well get combined with either wrinkled or green. Similarly, the roundness of a seed could also get combined with the green or the yellowness. So that means it's not necessarily that only the parental combinations will be there. There could be 4 type of combinations, 2 definitely parental and the 2 recombinants. So there are 4 types of phenotypes. If we were looking at the mathematical relationship, Mendel immediately could score the idea that a dihybrid cross was nothing but it was a square of the monohybrid. So that means indirectly we are talking about mono into mono. So that means if the relationship is of square, so that means the 3:1 ratio should also have a square. How do we make a square of a ratio? So in maths we will call it as 3 plus 1, which means the results would be 3 square plus 3 into 1 plus 1 into 1 plus 1 into 1, and which comes to 9:3:3:1. This is the basis. It's a very interesting way of depicting a checkerboard, and immediately we can find out if we are writing, because it's all maths, that if we are writing these particular 4 types of gametes in the correct sequence, then we take the larger triangle, it is made up of 9 different genotypic combinations, but then they will all constitute the same phenotype. Then the smaller triangle, which would be of 3, and still smaller of 3 and 1, which means this is the ratio of 3:1 whole square, which means 9, 3, 3, 1. Very interesting conclusion came when we talk about the genotypic ratio. You remember we had only 3 type of genotypes. They are called as the genotypic classes. 3, 1:2:1. And then what would happen to the genotypic ratio of a dihybrid? Naturally, like all other parameters, it also has to be a square. So the square of 1:2:1 would now have in place of 3 genotype classes, we would have 9, and it comes out to be as the square of 1:2:1, which is 1:2:4:1:2:1:2:1. So that means we have to utter this 9 times, and we have to be very careful. The sum total of this would be 16, whereas the sum total of 1:2:1 was 4. So in the coming episodes, we would again now look in details as to how to prepare the gametes, and I would also like to give you certain problems at hand. Thank you.
Thank you so much, sir, for this very interesting lecture with wonderfully prepared PowerPoint presentations for our aid from your side. And friends, as sir said that he would be taking it further in another lecture. So bringing this to a close, I would like to thank sir for his valuable contribution and time. Thank you so much, sir. And thank you, friends, for watching. Stay tuned. Have a great day.